Hi, my name is Abigail Meadows, and today we're going to be talking about mitosis. So in math, we know that multiplication and division are opposites, but in biology, when we're talking about cells, if we want to multiply, we first have to divide, and that's true through the process of mitosis. Before we get into the process of mitosis, I first want to talk about the cell cycle. Cells normally exist in a phase called interphase. This is where a cell spends the majority of its life, and some cells don't even divide, so those cells will be in a constant state of interphase. Cells here are maintaining themselves, and if they are going to divide, they're preparing themselves for that division through synthesis of their DNA, which is replicating their DNA. This is followed by the mitotic phase, which includes mitosis and cytokinesis. Prophase is the first stage of mitosis. You can think of it like an organized purse. You have a nucleus membrane that is still present, and within it you can see organized chromosome structures, which is not present during interphase. You can't see those individual chromosome structures. From prophase, we move into metaphase. Metaphase is where the nucleic membrane has gone away and all of the chromosomes meet in the middle of the cell. From metaphase, we go on to anaphase. In anaphase, chromosomes are pulled apart. Each side of the chromosome that's being pulled apart is identical. So you're creating two identical sides. But that starts really during anaphase. Those pieces that are being pulled apart, those chromosomes, are being pulled apart by telomeres on either end of the cell. So those telomeres that are pulling the chromosomes apart during anaphase continue to pull and bring us into telophase. In telophase, the chromosomes are beside their telomere that was pulling them apart, and they are starting to cleave from each other. So those two daughter cells are starting to cleave. At this point, we'll also see the nucleus membrane starting to form around the chromosomes once again. This brings us into cytokinesis, which is where the two daughter cells are separate. They're no longer cleaving, they've separated fully. We'd expect here to see cells that are identical to the original cell. So if that cell had organelles, these two daughter cells are also going to have those same organelles. It will look pretty much identical, again, to the original cell. These two daughter cells can start the process all over again, and that's why it's mitosis.